Hello students, how are you all? I hope so. You are keeping your health in good condition as well as mentally effective. Then you are enjoying your days. Am I right? Actually, I have taken a long break to come back to this YouTube channel, and henceforth I will be uploading my videos regular basis. Okay, please kindly give your cooperations and your support to my channel. So at least once in a day I will be uploading a video regularly. Okay, got it? So let's enter into the topic, and today I am going to discuss about molecular basis of inheritance. So molecular basis of inheritance. The chapter is 6 chapters in CBSE 12th standard textbook, as well as when we go for the state board also we are having this topic, molecular basis of inheritance. So before entering into this topic, what is molecular basis of inheritance? So எதனால இந்த டாபிக் நாம படிக்கணும்? So what is there behind this topic? So we might have studied Mendelian theories, am I right? So we have studied the classical genetics. When we studied the classical genetics, what is the abstract of this classical genetics? The Gregor Johann Mendel is the scientist. Okay, basically he was a teacher and then became a monk. Then he started to do many experiments in the Pisum sativum, and then finally he proposed a theory called Mendelian theory or classical genetic theory. So what this classical genetic theory says, okay, he has proposed 3 theories. The theory number 1 is a law of dominance. The second one is a law of segregation, law of independent assortments. So this theory explain the common concept is the factor is responsible for the transferring character from one generation to another generations. So where does this factor is available, where it is present? So when the further studies goes on to understand what is factor, then finally we have come across the term that is DNA. Today we are knowing about what is the genetic material, that is DNA. But in 18th and 19th century, no one knows what is genetic material. So at the time, the Gregor Johann Mendel has conducted many experiments and proved that the factor responsible for the transmission of the character from generation to generation, from the parent to offspring. The factor தான் ஒரு பேரண்ட்ல இருந்து ஆஃப்ஸ்பிரிங் போகுது. ஸோ ஒரு ஜெனரேஷன்ல இருந்து டிரான்ஸ்ஃபர் ஆகுது. பேரண்டோட எல்லா கேரக்டரும் எல்லா குணநலனையும் டிரான்ஸ்ஃபர் பண்ணுறது இந்த ஃபேக்டர் தான் சொல்றாரு. ஸோ இந்த ஃபேக்டர் வந்து ஒரு குறிப்பிட்ட கேரக்டரை ஒவ்வொரு ஜெனரேஷனுக்கும் கேரி ஓவர் பண்ணிருக்கு. ஓகே, ஸோ திஸ் ஃபேக்டர் இஸ் ஃபைனலி டேர்ம்ட் as a DNA. Today we know the genetic material is DNA, but that time nobody knows what is DNA. So every scientist, after studying the Mendelian theory, they started to investigate. They got an insight. So they wanted to enlighten themselves, okay, what is exactly transferring, which is exactly transferring the character. So thereby then we are going for the search for genetic material. Usually when we see the process, what is happening in our body, the DNA is the genetic material which is changed into mRNA. The mRNA is then producing the protein through the different process. So first the DNA is replicate itself, making many copies. Replicate. The DNA is replicated, then the replicated DNA undergoes the process called transcription. Transcription process. So in the transcription, this DNA, what the information is there, that is coded into mRNA. mRNA stand for messenger RNA. The mRNA is having the code for the particular amino acids. Okay, now this mRNA is in the form of coding. Then this mRNA undergoes the another process called translation. So what is happening in the translation? The coded informations are decoded. The coded information in the mRNA are decoded into amino acids. So number of amino acids, they join together and forms the protein. So who is helping for the protein synthesis? The ribosome. This ribosome carry the mRNA and then it finally decode it into the protein form. Got it? So this is an information. It's just like how this journalist will be doing their work. The journalist என்ன பண்ணுவாங்க, ஒரு பர்டிகுலர் ஸ்பாட்டுக்கு போவாங்க, அந்த இன்ஃபர்மேஷன் ஸ்பாட்டுக்கு. ஸோ எல்லாம் பக்கமாக அவங்களால எழுத முடியாது. ஒரு இன்ஃபர்மேஷன், ஒரு நியூஸை வந்து அவங்களால பக்கமாக எழுதி இருக்க முடியாது. அதனால் அவங்க ஒரு கோட் வேர்டு வச்சுக்குவாங்க. ஸோ ஒவ்வொரு சென்டென்ஸுக்கும் ஒவ்வொரு இதுக்கும் ஒரு கோடிங் வச்சுக்குவாங்க. இந்த கோடிங்கில் தான் எழுதி எடுத்துகிட்டு வருவாங்க. சரிங்களா? அந்த கோடிங்கில் எழுதி எடுத்துகிட்டு வந்து அதை டீகோட் பண்ணி அதை ஒரு இன்ஃபர்மேஷனாக நியூஸ் பேப்பரில் போடுவாங்க. The same thing happening in our body also. The DNA is replicated into multiple copies. The DNA, the parent DNA that's having the double stranded DNA. So double stranded DNA unwind and make into two single stranded DNA. Then synthesis of new DNA will take place. Afterwards, then replication overwhelms. Then it goes to the mRNA. The DNA is changed into mRNA. The mRNA, then it is with the help of the ribosomes, then it decodes the informations what is carried into the protein. So each and every code will be coding for the one particular amino acids. So this is, we call it a central dogma of the molecular genetics. Central dogma of the molecular genetics. This is a key point. So இதை சுற்றி தான் அந்த எல்லா ப்ராசஸும் நடக்குது. ஓகேங்களா? ஓகே, ஃபைன். ஸோ அப்போ what is DNA? Now we are how to identify the DNA. So search for the DNA. Search for the genetic material. How they are coming to the conclusion? Only DNA is responsible, not any other things. Maybe they may have the thought that maybe the protein will be responsible, or maybe the enzymes will be responsible, or the vitamins will be responsible. Whatever it is, then how exactly we are able to say that DNA is the genetic materials? So in order to prove this, a lot of experiment has been carried out. Okay, so in 1869, the Mesher, he has proposed that word, that is a nuclein. He has termed the word nuclein. The nuclein is nothing but it is a genetic material. To his term, to his understanding, he says it is a genetic material, but he did not say it is a protein or it is a enzyme. Am I right? So he has given the term nuclein, genetic material. But in 1926, all the scientists, they started to study the genetic material narrower and narrower to find out exact information, exact causes, exact reason, exact solutions and exact chemical substances, that is at the molecular level. So when they want to study at the molecular level, so they started in 1926. Okay, so in 1928, Griffith has conducted experiment. So it is the stepping stone of the genetic information identification or discovery. So he has given the stepping stone for the discovery of genetic material, that is the DNA is the genetic material. சரிங்களா? So Griffith experiment பண்ணாரு. This Griffith experiments we call it transformation principle. Transform. The something is transformed. So that something, how it is transformed, he did not know, but something is transformed. So which made to these transformations. So thereby, based on that, there we are calling it a transformation principles. So what he has done exactly in that experiments, he has conducted a series of experiments. Okay, he has taken, in order to conduct this experiment, he has taken the experimental sample, that is in streptococcus. Streptococcus pneumoniae. Streptococcus pneumoniae. The streptococcus pneumoniae, there are two strains. He has taken S strain as well as the R strain. S strain and R strain. Okay, what is S strain and R strain? How we are able to differentiate? என்ன சார் அதுல டிஃபரன்ஸ் இருக்கு அப்படினு பாக்கும்போது S strain இருக்கு இல்லீங்களா, the surface of the S strain bacteria is having the mucus layer. So the mucus is nothing but the polysaccharide, mucopolysaccharide. So அதனால இதனுடைய surface வந்து ரொம்ப ஸ்மூத்தா இருக்கு. But in case of R strain bacteria, they don't have the mucus polysaccharide. So thereby, no mucus. So what happens? The surface is very rough. Okay. So Griffith has taken two strain of streptococcus pneumoniae, and one strain is an S strain, another strain is an R strain. Okay, so when he has taken the S strain, is having the mucus polysaccharide. Because of that, the S strain is very smooth. So that's why we call it a smooth strain. Do you understand what I am saying? Got it. Now he is going to do the experiments. Please watch carefully what is going to happen. Okay, so he has taken the S strain and injected into mice. The mice is a well-known experimental animal. So whatever medicines, whatever chemical substances, drugs we want to use it, we have to testify in the mice, then only we can use it. It can be commercially used. Okay, so when it is injected into the mice, to his observation, the mice die. He was wondering how it is. So what made it to die? Okay, then he has started another experiments, that is, he has taken R strain. So R strain educated, he is again doing the injections, injected into the mice, Okay, the mice is alive. What a miracles. So when he has injected the S strain, the mice die. When this R strain is injected into mice, the mice is alive. Okay, then what is the problem? Still he is not very clear with his idea. So he has come to the conclusion one thing, the S strain is having something, the pathogenicity. So S strain is a virulent. He call it a virulent. Okay. So since this S strain cause pneumonia in the mice after the injections and leading to the death of the mice, so thereby he call this S strain is a virulent, having the pathogenicity. What is pathogenicity? The intensity of causing disease in the organism. The intensity of causing disease in the organism, we call it pathogenicity. We call it pathogenicity. Pathogenicity. புரியுதுங்களா? Got it? Okay. R strain is not causing any pneumonia. I mean, because this is a streptococcus. Pneumonia can cause pneumonia in the organism. But Austin does not cause pneumonia in the mice. So that's why the mice is alive. So thereby this Austin is termed as a non-virulent. Non-virulent. Non-virulent in solid. Okay, still you want to combine both the things. So what he has done, he has taken a stain and killed it. So he has introduced the killed S strain into mice, injected into the mice. Okay. So when it is injected into the mice, the killed one, the mice is alive. The mice is alive. Okay. When he has taken the S strain as an alive, that is, and it is able to cause the diseases in the mice and causing the disease in the mice, leading to the death of the mice. But when he has taken the S strain killed, heat killed it. That's we call it an heat killed S strain. When he introduced heat killed S strain into the mice is alive. The mice is not dead. Okay, then you want to combine again. The heat killed S strain plus R strain. Heat killed S strain and plus R strain. So this mixture is injected into the mice. To our sadness, the mice is dead. The mice die. The mice die. So in the beginning itself, he has done that S strain is a virulent and R strain is a non-virulent. Now what happens when we has taken the heat killed S strain and R strains? So something changed R strain into virulent form. And the something it is absolutely the S strain. So S strain ஏதோ ஒரு substance R strain க்கு போய் அத வந்து pathogen அதாவது pathogenicity மாத்திடுது. அதாவது S strain ஏதோ ஒரு substance R strain க்கு போயிட்டு R strain virulent மாத்திடுச்சு. This is we call it an transform. The heat killed S strain transformed R strain into virulent. Got it? Clear? Okay. So far, something, the genetic materials is responsible, or here we call it the mucopolysaccharide, is responsible for causing the virulent or causing the pathogenicity. Still not satisfied. Still not able to find which is exactly causing, whether that is a protein or whether it is an enzymes, whatever it is, and still yes. Chase method. This two scientists. Okay, in 1953, around 1952, they conducted experiments called Chase experiments. So what is why they have to conduct Chase experiment? And they wanted to conclude whether the protein or whether the enzymes or RNA or DNA causes pathogenicity, or which is transferring the genetic information. So they have taken two radioactive isotopes, that is phosphorus 32. Okay, there are another radioactive isotopes, that is in sulphur 35. Okay, these two are radioactive isotopes. Radioactive isotopes. I hope so you are understanding. Okay, so they have taken two radioactive substances, one is phosphorus and another one is in sulphur. Okay, fine. So first what they have done, they have taken the culturing plate. So this is an culturing plate. Okay, so they have taken the culturing plate 1 and culturing plate 2. Before that, they have taken bacteriophage. Okay, so this bacteriophage is allowed to infect bacteria. Allowed to infect bacteria. We know that bacteriophage is a virus. Okay, so the virus which infect the bacteria. The virus which infect the bacteria, that is we call it a bacteriophage. The bacteriophage is having encapsulation. They are having the head regions as well as the tail regions. The head regions is encapsulated with a covering. I mean the encapsulation is nothing but the cover. Okay, inside the encapsulation we are able to find out the genetic material. Usually what happens when the bacteriophage come and infect the bacteria think that this is our genetic material. So what it will do? It will start to replicate the bacteriophage DNA. It will not recognize it is a different one. So the bacteriophage once infect and the DNA of the bacteria and the bacteriophage la irukra and the DNA வந்து bacteria-ல இருக்கிற அந்த DNA கூட integrate ஆயிடும். ஒண்ணுக்கு ஒண்ணா சேர்ந்துடும். ஏன்னா பாக்டீரியா என்ன நினைக்குது, ஓகே இது நமக்கு கரெக்டான ஜெனடிக் மெட்டீரியல் தான், நம்ம ஜெனடிக் மெட்டீரியல் தான், அதனால நம்ம ஈஸியா இதை மல்டிப்ளை பண்ணலான்ட்டு இட் இஸ் டூயிங் ஆஸ் யூசுவல் ஒர்க், doing the replication, transcription and translation. So replication பண்ணும்போது then this bacteriophage virulent DNA are multiplied. அந்த bacteriophage உடைய DNA வந்து மல்டிப்ளை ஆயிடுது இந்த பாக்டீரியால. சரிங்களா? So இதை எடுத்து அவங்க வந்து பெட்ரி டிஷ் இருக்கு, அந்த பெட்ரி டிஷ்ல அந்த bacteriophage எடுத்து இந்த bacteriophage-ல P32 இந்த ரேடியோஆக்டிவ் சப்ஸ்டன்ஸ் இருக்கு. இன்னொரு பெட்ரி டிஷ்ல ஐ மீன்ஸ் கல்ச்சரிங் பிளேட்ல S35 இருக்கு, S35 இருக்கு. சரிங்களா? S35. So இப்போ இந்த bacteriophage யூஸ் பண்ணி இந்த bacteriophage இந்த கல்ச்சரிங் மீடியத்துல grow பண்றாங்க. Why they have to do it? This bacteriophage should have the incorporated substances, radioactive substances. புரியுதுங்களா? What this Hershey's மெத்தட்ல யூஸ் பண்றாங்கன்னா bacteriophage எடுத்து பாக்டீரியாவை இன்ஃபெக்ட் பண்றாங்க. சரிங்களா? சோ அது இன்ஃபெக்ட் பண்றதுக்கு முன்னாடி என்ன பண்றாங்கன்னா இந்த bacteriophage எடுத்து அந்த ரேடியோஆக்டிவ் சப்ஸ்டன்ஸ் இருக்கு இல்லையா, அந்த ரேடியோஆக்டிவ் சப்ஸ்டன்ஸ் கல்ச்சரிங் பண்றாங்க. ரெண்டு விதமான ரேடியோஆக்டிவ் சப்ஸ்டன்ஸ் எடுக்கறாங்க. ஒன்னு வந்து phosphorus 32, இன்னொன்னு வந்து sulphur 35. So two different culturing plateல இத வந்து கல்ச்சரிங் பண்றாங்க. Okay, culturing plate 1ல bacteriophage is introduced and this when it's introduced into the culturing plate 1, the culturing plate 1 is having phosphorus 32 isotopes. So now this phosphorus 32 isotopes is incorporated in the genetic material of bacteriophage. The phosphorus 32 வந்து உங்களுக்கு bacteriophage உடைய genetic material incorporated ஆகுது. That means it's entering into the genetic material. சரிங்களா? அதே சமயத்துல கல்ச்சரிங் பிளேட் 2ல திரும்பி bacteriophage போடுறோம். அந்த bacteriophage-ல this sulphur 35 also incorporated into the bacteriophage. இப்போ நம்ம क्वेश्चंस என்னன்னா இதுல encapsulated headல வந்து phosphorus இருக்குமா இல்ல encapsulated அது genetic materialல phosphorus இருக்குமா என்றதான். யூசுவலா DNA ன்னு பாத்தீங்கன்னா DNA ல phosphorus, DNA is having the phosphate group. சோ அப்ப இந்த phosphorus the DNA ல இருக்கணும். சரிங்களா? அதே சமயத்துல இந்த சல்ஃபர் என்றது is a protein containing amino acidsல வரும் இல்லீங்களா? சோ amino acidsல வரும்போது இது கண்டிப்பா இந்த protein capsule தான் இருக்குன்னு ஒரு predictions இருக்கு. ஆனா அது confirmation ஆகல. So what this Hershey and Chase method they are doing, they are taking the radioactive isotopes. First they have taken the bacteriophage, okay, incorporated into the culturing plate which contains an isotopes. 25. Okay. After that, this incorporated bacteriophage is allowed to infect bacteria. Bacteria infect பண்ணுது. சரிங்களா? After infections, so what happens then? Blending. அதாவது grind பண்றோம் அதை. அது ஃபுல்லாவே நம்ம grind பண்ணி centrifugeல போடுவோம். Then we are going to keep it in the centrifuge. Okay, first what we are doing, we are taking the bacteriophage and this bacteriophage is incorporated. Some of the bacteriophage is incorporated into radioactive isotopes P32. Some of the bacteriophage is incorporated into radioactive sulphur 35. Okay, after the incorporations of the bacteriophage, then we are transferring this bacteriophage into the bacteria to infect. So after the infection, what happens? The bacteria started to multiply the DNAs and as well as the protein molecules. I mean the capsule. The bacteria started to multiply the genetic material as well as the capsule. Okay. So that is blended. That means that is extracted and that will be sent to the centrifuge for the centrifugation purpose. So in the centrifugation, what happens? The high molecular density, the molecules which is having higher density will settle at the bottoms. The molecule which is having the lower density, it will be at the top. Okay, which is having the medium molecular weight, it will be in the middle of the centrifuge. Okay, so when we have sent this blending extract to the centrifuge and we are able to separate out two things, able to observe two things. Okay, the genetic material, when we analyze the genetic material, shows P32 radioactive. Genetic material shows P32 radioactive substances. That capsule shows S35. By this we are able to confirm. So the genetic material is having phosphate group. It will be the DNA. The genetic material is in acid form. The genetic material is in acid form. Okay, nucleic acid. The genetic material will be nucleic acid. Okay, so the capsule which is absorbed, which is incorporated, that shows that S35. So thereby it is a protein. So now Hershey and Chase has given the solid confirmation that the DNA, the genetic materials only carry the information from one generation to another generation, not the protein, not the enzymes. You understand? Okay, so who has given the solid information? The Hershey and Chase method is the radioactive isotopes, using the radioactive isotopes. Okay, come back to the DNA. So far we have seen the search for genetic material. Am I right? The search for genetic materials. Okay, so now come back to the DNA. What is DNA? What is the full form of DNA? We call it an deoxyribonucleic acid. We call it deoxyribonucleic acid. So, got it? So this is the nucleic acid, is the genetic material. So the nucleic acids are two categories. One is an RNA, another one is an DNA. RNA and DNA. Most of the eukaryotic organisms, they have the DNA as their genetic material, and the virus will have RNA as the genetic material. What is RNA? That is an ribonucleic acid. Ribonucleic acid. This is, we call it an RNA. Okay. So the genetic material is a polymers of nucleotide. We call it DNA is a polymers. DNA is a polymers of nucleotide. Nucleotide. Sir, what is nucleotide? Polymers of nucleotides. So this polymers of nucleotides form the DNA structure. Sir, what is nucleotide? Okay, so ribose sugar. The ribose sugars linked to the nitrogenous base. Ribose sugar linked to the nitrogenous base. Nitrogen bases are adenine, guanine, cytosine, thymine, and uracil. These are all nitrogenous base. Okay, so ribose sugar linked to the nitrogenous base through N-glycosidic bond. How it can be linked? Through the N-glycosidic bond. I will be explaining how it is linked. Okay, so just keep it in order to find out the nucleotide. So ribose sugar linked to the nitrogen base and forms nucleoside. Okay, so to the nucleoside, if phosphate is added, inorganic phosphate added, then we call it a nucleotide. I hope so now you will understand what is nucleotide. So nucleotide it is having the components of ribose, nitrogen base and phosphate. Ribose, nitrogen base and phosphates. This is the nucleotides. So the DNA is composed of polymers of nucleotide. How it is composed? How this polymer of nucleotide is joined together? So we have studied the Griffith experiments. The Griffith experiment says that when the S strain, heat killed S strains combined with R strain, the R strain became virulent. And he says, so some materials, maybe the polysaccharide or maybe the proteins or maybe some other enzymes can cause the transformation of R strain into virulent form. Okay, so but exactly this Griffith did not know what is the biochemical nature of this strain. So thereby, followed by him, there was another experiment conducted by that Oxford Avery et al. So McCarthy, the scientists. So before they start their experiments, before their discovery, before their experiments, the thought was like this. So the genetic material may be the protein. So that was the thought before the Avery et al. Experiments. And this Avery et al. Experiments, it's followed different steps. Okay, first steps, what he has done, he removed the Avery et al. They removed the polysaccharide capsule from the S strain, that is heat killed S strain and R strain. When he removed, what happens? The mice died. So removed the polysaccharide capsule from the heat killed S strain and to the R strain. Okay, so what happened? The mice died. So again he has conducted another experiments. He removed the protein. They removed the proteins from the heat killed S strains and with the R strains. So after removing protein also, the mice is dead only. The mice is died. Okay. Then he has gone to the next level, that he added a protease enzymes to the heat killed S strain as well as to the R strains. The mice still died. And he is getting a little more enthusiasm. So what will happen to the next level? So thereby, so he has carried out another experiments, that is in step 4, he removed RNAs from heat killed a strain and or strains. So to his surprise, and still he is able to get it, the mice died. The mice died. So what will be the answer? This is going on. First he added that he has removed the polysaccharide from the heat killed S strains with an R strain. The mice died. And the second he removed the protein from the same combinations. Still the mice died. And third he has removed the protease. He added the protease and which can degrade the protein, and still the mice died. So then finally he has gone to the next level, that is when he added the RNA. He added RNAs. So when he added the RNAs to the heat killed S strains with that R strain, what happens? The mice still died. And still that the mystery is not revealed. So he has gone to the next level. In the step 5, he added DNAs which can degrade the deoxyribonucleic acid. He added DNAs to heat killed S strains with R strains. Okay. So to his astonishment, exactly the mice survived. The mice survived. Oh, thanks to his experiments, and he has finally come to the conclusion nothing is genetic material except this DNA. No protein, no polysaccharide, no RNAs, and no enzymes. Nothing is responsible for transformation of R strain into virulent when that heat killed S strain is added. So this heat killed S strain transferred DNA to the RNA. So thereby this RNA, heat killed S strain transferred DNA to the R strain. So thereby the R strains became virulent. It can cause pathogenicity. So DNA is the genetic material. Okay. புரியுதுங்களா? ஸோ ஏவரி எட்டால் என்ன பண்ணுறாங்க, அந்த கிரிஃபித் எக்ஸ்பெரிமெண்ட்டை ரீடிசைன் பண்ணுறாங்க. ஸோ கிரிஃபித் எக்ஸ்பெரிமெண்ட்டில் என்ன நடந்துச்சுன்னா, அவர் 2 விதமான ஸ்ட்ரெயின் எடுக்கிறாரு. ஒரு ஸ்ட்ரெயின் வந்து எஸ் ஸ்ட்ரெயின், இன்னொரு வந்து ஆர் ஸ்ட்ரெயின். இந்த எஸ் ஸ்ட்ரெயின்ன்றது உங்களுக்கு அந்த பாலிசாக்கரைட் கேப்சூல் இருக்கிறதால ஸ்மூத்தாக இருக்கும், அதனால அது ஸ்மூத் ஸ்ட்ரெயின்னு சொல்கிறோம். இந்த ஆர் ஸ்ட்ரெயின்ன்றது பாலிசாக்கரைட் கேப்சூல் இல்லை, அதனால இது ரஃபாக இருக்கு, இந்த ஆர் ஸ்ட்ரெயின் என்றோம். சரிங்களா? ஸோ ஃபர்ஸ்ட் என்ன பண்ணேன்னா, அந்த கிரிஃபித் எஸ் ஸ்ட்ரெயினை மைஸில் இன்ஜெக்ட் பண்ணும்போது இந்த மைஸுக்கு நிமோனியா வந்து இறந்து போயிடுச்சு. சரிங்களா? திரும்பி ஆர் ஸ்ட்ரெயினை மைஸில் இன்ஜெக்ட் பண்ணும்போது இந்த மைஸ் வந்து நிமோனியா இல்லாமல் அலைவாக இருக்குது, சர்வை ஆகுது. ஸோ அடுத்த எக்ஸ்பெரிமெண்ட் என்ன பண்ணேன்னா, இந்த எஸ் ஸ்ட்ரெயினை வந்து அதிகமான ஹீட் அப்ளை பண்ணி அதனுடைய விருலண்ட்டை வந்து குறைச்சிட்டாரு. ஓகே. ஸோ he made எஸ் ஹீட் கில்டு ஸ்ட்ரெயினை மாற்றிட்டாரு. ஸோ ஹீட் கில்டு ஸ்ட்ரெயினை மாற்றி அதை மைஸில் இன்ஜெக்ட் பண்ணும்போது அந்த மைஸ் அலைவாக இருக்கு. ஸோ இட் இஸ் அன் அலைவ். ஸோ ஃபைனலாக என்ன பண்ணுறாரு, இந்த ஹீட் கில்டு எஸ் ஸ்ட்ரெயினை வித் காம்பினேஷன் ஆஃப் ஆர் ஸ்ட்ரெயின் கூட கம்பைன் பண்ணி மைஸில் இன்ஜெக்ட் பண்ணுறாரு. இந்த மைஸ் இறந்து போயிடுது. ஸோ இது எப்படி பாசிபிள்? அப்போ அவர் எஸ் ஸ்ட்ரெயின்ன்றது விருலண்ட்னு கால்குலேட் பண்ணியிருந்தாரு, ஆர் ஸ்ட்ரெயின்ன்றது நான் விருலண்ட். அது எப்படி இந்த ஹீட் கில்டு எஸ் ஸ்ட்ரெயின் எப்படி ஆர் ஸ்ட்ரெயின் கூட கம்பைன் ஆகும்போது அது எப்படி விருலண்டாக மாறுது என்ற ஒரு கன்ஃபியூஷன் வந்துச்சு. மேபி இதில் இருக்கிற பாலிசாக்கரைடாக இருக்கலாம், இல்லை இதில் இருக்கிற அந்த புரோட்டீனாக இருக்கலாம் என்ற ஒரு அசம்ஷனில் தான் இருந்தாங்க. ஸோ அந்த அசம்ஷனை அவருக்கு அந்த கிரிஃபித்துக்கு வந்து அந்த பயோகெமிக்கல் ஸ்டடீஸை பற்றி தெரியல ப்ராப்பராக, அதனால் அவர் அதை விட்டுட்டார். ஸோ அதை ஃபாலோ பண்ணி வந்தவங்க வந்து தட் இஸ் அன் ஆஸ்வர்ட் எட்டால், ஏவரி எட்டால், அவங்க என்ன பண்ணாங்கன்னா, தே ஹவ் டேக்கன் another level பயோகெமிக்கல்ஸ் studies பார்க்க ஆரம்பிச்சாங்க. இந்த biochemical studiesல they have extracted the biochemicals from the heat killed strain. So heat killed strainல இருந்து அந்த biochemical extract பண்ணிட்டாங்க. சரிங்களா? இப்போ extract பண்ணிட்டு அதை வந்து ஒவ்வொரு ஸ்டேஜஸா அப்ளை பண்ண போறாங்க. ஃபர்ஸ்ட் ஸ்டேஜ் இந்த ஸ்டெப் 1ல என்ன பண்றாங்கன்னா இந்த பாலிசாக்கரைடை கம்ப்ளீட்டா ரிமூவ் பண்றாங்க. அந்த S killed ஸ்ட்ரெயின்ல இருந்து R ஸ்ட்ரெயின்ல இருந்து அந்த பாலிசாக்கரைடை கம்ப்ளீட்டா ரிமூவ் பண்ணிடுறாங்க. ஸோ கம்ப்ளீட்டா ரிமூவ் பண்ணும்போது என்ன நடக்குதுன்னா மைஸ் இஸ் ஸ்டில் டெட். சரிங்களா? அது இறந்து தான் போகுது. அதுக்கு அடுத்தது என்ன செய்யறாங்கன்னா அதில் இருக்கிற அந்த புரோட்டீனை வெளியே எடுத்துறாங்க. அந்த ஹீட் கில்ட் S ஸ்ட்ரெயின் R காம்பினேஷன்ல அதில் புரோட்டீனை வெளியே எடுத்து பார்க்குறாங்க. ஸ்டில் த மைஸ் இஸ் டெட். சரி, அடுத்த ஸ்டேஜுக்கு போறாங்க. என்ன செய்யறாங்கன்னா இப்போ புரோட்டியேஸ் என்ற ஒரு என்சைம் வந்து ஆட் பண்றாங்க. இந்த புரோட்டியேஸ் உடைய வேலை என்னன்னா புரோட்டீனை வந்து டீக்ரேட் பண்ணும். So when they added the protease enzyme to the heat killed S strain and R strain, what happens to that? Then that mice still died. So திரும்பி அது இறந்து தான் போயிடுது. So then finally he has gone to the next level. He has added the DNase. DNase என்ற enzyme அந்த heat killed S strain with R strain கூட ஆட் பண்ணும்போது what happens no? Then that mice survive. அப்போ என்ன நடந்துக்குது? இந்த டிஎன்ஏஸ் இருக்கு இல்லையா, இந்த டிஎன்ஏஸ் என்சைம் வந்து டிஎன்ஏ வந்து டீக்ரேட் பண்ணிடும். அப்போ அது டீக்ரேட் ஆயிடுச்சுன்னா ஆட்டோமேட்டிக்லி இட் லாஸ் இட்ஸ் பேத்தோஜெனிசிட்டி ஆர் இட்ஸ் விருலன்ஸ். ஸோ தேர் வித் த மைஸ் சர்வைவ். ஸோ இவங்க தான் இந்த ஏவரி எட்டால்ஸ், இவர் தான் இந்த ஆக்ஸ்ஃபோர்ட் ஏவரி எட்டால்னா அப்போ டிஎன்ஏன்றது ஒரு ஜெனட்டிக் மெட்டீரியல் என்று கன்க்ளூஷன் கொண்டு வராரு. So when this conclusion is done and it is not completely accepted by most of the scientists, so thereby the Hershey's chase method is another revolutions in the finding out of the DNA is the genetic materials. So Hershey's chase method, he has taken the bacteriophage. Okay, the bacteriophage is cultured in the isotopes. He has taken the two isotopes form. One is in phosphorus 32, another one is in sulphur 35. So he has cultured in two culturing plate. So some of the bacteriophages added to the culturing plate 1, which is containing phosphorus 32. Another culturing plate 2, which is to which the bacteriophage is added, and this culturing plate 2 contains the sulphur 35 isotopes. So after that, these isotopes are incorporated in the bacteriophage. Now this bacteriophage is allowed to infect the bacteria. After the infection, the bacteria multiplies more and more copies of the radioactive isotopes genetic materials. When it is blended and that means extracted, this extract is used in the centrifuge techniques in order to identify which is having the high molecular weight, which is having the low molecular weight, and in order to separate out which substance is having phosphorus 32, which substance is having sulphur 35. To his surprise, he is able to identify the genetic material incorporated phosphorus 32, then the protein capsule incorporated sulphur 35. So thereby he has concluded DNA is the genetic material and it is the nucleic form. It is in acid form. So in the next sections, we will be discussing about how far the structure of the DNA, then the length of the DNA, how this DNA is tightly packed into the nucleus. Thank you so much. Have a nice day.